Hello, my name is William Hines, and I'll be going over the Yersinia Pestis disease, also known as the Black Death. And it was also in three of the deadliest pandemics, but we'll be talking about the Black Death portion of it. So let's get straight into it. So the Yersinia Pestis is the disease that led to Black Death from 1346 to 1353. It's considered one, if not the deadliest plague of all time. It's a gram negative, so that means it's dangerous because the outer membrane is often hidden by a capsule, and it just acts as a camouflage by hiding the antigens. And it's also non-modal, meaning this it lacks the ability to withstand a structure. It's a rod-shaped coccobacillus bacteria with false spores, and it also is a facultative anaerobic organism that can affect humans. All that means is that an organism that makes ATP by aerobic respiration if oxygen is present, and they killed around 25 million during the Black Death, and it's technically still around today. And I would like to talk about the other plagues it was involved in. Three of the deadliest pandemics recorded in history were caused by this single bacteria. The plague of Justinian, which was the very first one that was involved in, which was carried over the Mediterranean Sea from Egypt with rodent fleas, and they catch the wild on black rats who snacked on the grain on the ships. It killed from 30 to 50 million people, and it spread across Europe, Asia, North Africa, and Arabia. And I don't know why it's not talked about as much, but it was the deadliest one. And then the most recent one, the third one, is the Great Plague of London. Sealing up the sick was a nickname because London had to seal up the sick in their homes and bury the dead in mass graves, which they had to do that for the most part for the Black Death. And it may have been the only way to bring the outbreak to an end, and it ended up killing 100,000 Londoners in under 7 months. And we'll also be talking about the three main forms of it, which is pneumonic, septicemic, and bubonic later on in the slides. Now let's get to how it started. The plague arrived in Europe in October 1347 when 12 ships from the Black Sea docked at the Silicon port of Messina. Most of the sailors on the ship were already dead, and those that were alive were gravely ill with black boils that oozed and pus. And they tried to order the quote unquote death ships out, the people that were on it, but it was already too late. Then start over the next 5 years, the Black Death would kill more than 20 million people in Europe. Now, the Black Death slash plague first started during the plague of Justinian, as I just talked about last slide, spreading the most in the Himalayan mountains of South Asia in the 1200s, but ended up staying around, and it just got bigger once the rats got back on the ship. What I just talked about in 1347. And since living conditions were often cramped and dirty, humans lived in close contact with rats, and the Europeans were scarcely equipped for the horrible reality of the Black Death, quote unquote. And men and women alike, there were certain swellings that were called plague boils, and they would just rot on their skin, and they would look very nasty. And also, the black rats were the most common at the time, like common type of rats, and already carried the bacteria, and they'll just have fleas usually on them anyways that just would jump around onto humans and bite their skin and also give them the disease. Now onto the symptoms. People who have bubonic or septicemic plague, as we will talk about in a second, can develop Yersinia pestis infection in the lungs. Other symptoms are high fever, chills, headaches, chest pain slash rapid breathing, severe shortness of breath and cough that might bring up blood. And back then, without proper treatment, the disease can quickly lead to death. And they weren't even close to as medically advanced as we are now. So usually back then when you got it, it was already over basically. Oops, let me get back. All right, on to the three main forms of Yersinia. The bubonic plague attacked the lymphatic system, causing swelling in the lymph nodes. And if untreated, the infection can spread to the blood or lungs. And 1 to 7 days after the exposure, flu-like symptoms also start to develop with the bubonic plague. These symptoms include fever, headaches, and vomiting. And as you can see right here, how it goes throughout the body, the swells and the gangrene on the fingers. and also the seizure. Now onto septicemic, which is a life-threatening infection of the blood, mostly spread by bites of infected fleas, like I just talked about, W in the rats. Like other forms of gram-negative sepsis, it can cause decimated intravascular coagulation. And what that means is it's a condition in which blood clots form throughout the body, blocking small blood vessels. And it's always fatal when untreated. As you can see, they have cerebral blood clot right here, and it's most likely going through the body right now. And last, which is the most deadliest and easiest to get infected by because it's airborne, is pneumonic. Pneumonic, pneumonic plague is a severe lung infection caused by bacterium Yersinia. And as you can see in the x-ray right here, the person that has it in their lungs. Now, onto the doctor that solved the bacteria, Alexander Yersin. Alexander was a dual national Swiss and French physician bacteriologist. While he was a medical student working in Paris, he was trying to discover the exotoxin, which is a toxin released produced by the diphtheria bacillus. So he already had experience with finding different bacteria. So years after he graduated, he was sent to Hong Kong in 1894, way after the Black Death, to find the cure for the outbreak of the bubonic plague. And it says he is remembered as the co-discoverer of the bacillus for the bubonic plague. But when I try to find the other person that helped him, I couldn't find anyone. So I just wrote him down. And he isolated from buboes the causative bacillus Yersinia Pestis, which made it easier for him to find the bacteria and the problems with it. And he also developed vaccines and interserum for both men and animals. And since he did all this, it was later named Yersin in honor of him. Now onto the rodents. And also more about the bacteria. The people knew that the bacteria traveled from person to person through the air, which many scientists already suggested that they knew. But as well through the bite of infected fleas and rats. Many of them didn't know that at first when they first started. So let's talk about first the rats. Alexander also demonstrated for the first time that the same bacillus was present in rodents. And Yersinia was spread by the bite of infected fleas. And the fleas would come off the rats, as I said already, and frequently carried by rats, causing the bubonic part of the plague. And both of the pests could be found everywhere in medieval Europe, but they're particularly at home aboard ships of all kinds because the grains will already be on the ships and those travel with it, which is how the deadly plague made its way throughout the country and Europe city port one after another. And that's also how it spread throughout the whole world after going port to port with ships. And many European cities were already dirty and already fleeing with rats as they didn't have the best hygiene or trash transportation system back then. Now, onto a big question too. After doing more research, some experts don't think it was actually rats. They think that it was actually humans that caused the outbreak at first. A model done by the University of Oslo modeled the 3 transmission routes for the disease. Rats, as we just talked about, airborne, which is the pneumonic part of it, and human fleas and lice. They think it's human fleas and lice because they didn't have a great hygiene back then. And lice would just jump off from, say, a rat or something like that. And the fleas already came from the rats. And the lice would just go up through their hair usually or through the body. And also then using analysis with the mortality rates with the outbreak, the model suggests the spread of the outbreak was mainly attributed to human fleas and body lice. I think it was mainly rats, but this is also a great point that experts brought up. So it can also be this part too. Now, how it ended, the Black Death died out. It died out by simply being too deadly to persist, meaning that evolutionary theory tells us that a pathogen that kills all of its victims will eventually just run out of victims. So this is how quarantining was invented. The uninfected will remain in the homes and leave when necessary. This will help because the infected will just stay in their homes. Most of them probably sadly ended up dying, but it helped everyone else around them out because it will just stay in their homes and only affect themselves basically. And it won't spread out. That's what we do now with COVID-19. Well, what we did like 6 or 7 months ago, just quarantine. And that's probably the best way to end the epidemic. But it also could have just ran out of victims near the end too, as we talked about earlier with the Justinian plague. I meant the London plague, my fault. Now onto the cure to it. They have some bizarre cures. The ones that helped the most as I searched up was rubbing onions, herbs, and drinking vinegar. Now, the chopped up snake, if they have available in the black boils, I don't think that worked either. Cutting up a pigeon and rubbing it over affected body. And a couple other cures they used were eating crushed minerals, arsenic and mercury, or even 10-year-old tentacles out. They thought putting animals on the wounds would help it the most. I don't know why. And nowadays, things are certified by CDC, which is the Center for Disease Control. And we have many advanced antibiotics now, as we already have like vaccines for COVID-19 because we have more advanced technology now to help with medicine. So that's good. And here's why we're excited. Thank you.